From Microbe TV, this is Office Hours for Wednesday, June 7th, 2023. I'm Vincent Racaniello, and there's nobody to welcome. It's just me. Hello, everyone. Welcome to the Wednesday evening corner of the internet, where you can find out as much about viruses that I know. And I know some things. I don't know everything. But this is a good place to go. And hey, where else are you going to find a virology professor to hang with you for a couple of hours? I've been running around, as you know. I'll tell you a little bit about it, but I want to thank all of you for coming tonight. 51 so far. Let's see if we can get that into the 200s. Probably. I want to thank our moderators. We have in the house Vanity Nutrition. We have Les. We have Steph. We have Tom, we have Andrew, and that looks to be the crew for tonight. It's a great crew. Thanks for joining us and protecting our vulnerable viruses. Weird business here in the U.S. tonight. Got off the train from Washington, and as I was pulling in, look outside of it, it's yellow. I said, what are the windows tinted? It's yellow. Holy cow. And I got out and it smelled of smoke. Really interesting. It's still a little bit yellow and smoky, but much less tonight. What else has been happening? Yeah, I got back from another trip to the D.C. area, this time to do a TWIV with Rich Condit at USAMRID, United States Army Medical Research Institute for Infectious Diseases. And I've heard of it, of course, but really didn't know what it was, and it turns out to be eye-opening. Let me show you some pictures first. Here is the TWIV with, this is John Dye here, and this is Crystal. What's her name? Let me dig up her name. We can't be not having people's proper names. Crystal Burke. So they're both civilians. There's, of course, me and Rich. A little spread out, but that's, they did it for us. And here we have Jay Hooper, who's another civilian. And there is Todd Bell, who is about to be a civilian, actually, because he's just put in 20 years in the military. And, you can retire on full pension after 20 years, and you can get another job and still collect the pension. So that's what he's doing. He's going to Iowa State, where he's going to be a veterinary pathologist. So he's a veterinarian, and he's been doing pathology at USAMRID. So as you may know, in the U.S. military, you spend 3 years at any one base, then it's time to move on. So he's been all over the place, and he has been at USAMRID 3 different times. But he's also been in Iraq, he's been in Japan, he's been all over the place, doing amazing stuff with infectious diseases. So it's a mix of Army and civilians, and it's a huge base. Okay, it's got USAMRID is just one building. There's also the National Cancer Institute, which is a huge thing, which is in that fort as well. There's regular military folk who live on base. There's the NIH BSL-4. So USAMRID has a BSL-2, 3, and 4, several BSL-4s. Then there's the BSL-4 from NIH on campus, which is called the Integrated Research Facility. Then there's another BS4 in another building called NBAC. Don't ask me what it stands for. And anyway, we got to visit USAMRID, and it was cool. And I just learned that, they do research. In this place, they do research on the high-profile viruses and bacteria, like Ebola and Lassa and Marburg and Hantas and Nipah, because the servicemen and women are at risk for encountering them, so they want to have countermeasures and diagnostics. And of course, when COVID hit, they were helping everybody So they spill over into the civilian population to help as well. Anyway, you're going to hear all this in the TWIV, which will come out on Sunday, because they talk about it. And I mean, we were blown away. I didn't know any of this stuff. And you know what's very interesting? You know, you figure, oh, the military has trillions of dollars. But actually, they don't pay for any of this research. These guys have to write their own grants to get money. The only thing the Army pays for is the building and the air and the electricity and so forth. So very interesting. Anyway, one more picture from that. Let's shrink it down a bit. This was having after dinner in Fort Detrick. So, yeah, Fort Detrick is the name of the town. It's also the name of the base. And it's Fort Detrick is your typical, Maryland, Virginia colonial town. And this is Rich, of course. That's John Dye, who you might remember was on a TWIV where we talked about the consortium making monoclonal antibodies with Kartik Chandran and a bunch of others. And that's Todd Bell. So Todd Bell got this whole thing going a year ago. He invited us to go there. Anyway, so that's that. And what am I doing next week? This is very interesting. I am going to Philadelphia, University of Pennsylvania, where there's a little symposium on viruses. I'm going to give a talk on can polio be eradicated, and there are 3 other talks. And then we're going to do a TWIV with me and Susan Weiss and 3 other guests, one of whom is a retired FBI agent. He worked in the FBI for 25 years. And maybe he'll shed some light on what it is that the FBI thinks they have about SARS-CoV-2 origins. Now, he told us he doesn't know anything, but he could tell us, what they might be looking for. I want to ask him, what does it mean to have low confidence? Is that like we should just ignore it? Or what? Anyway. So that's next week. And for that reason, that's Wednesday. I'm not going to do a live stream because I'll probably be having dinner with them and it'll bump into that. All right. And then the following weekend, I'm going to Houston for ASM microbe, and I'm going to do a TWIV and TWIM on Sunday. And the TWIV will be with Eddie Holmes. So that'll be fun. Come back on Monday. And then the following Friday, I go to Georgia State for ASV. So those are and at ASV, I don't know if I'll do a office hours. I'll think about it. But that's the schedule for the next couple of weeks. We're staying busy. And so for all of June, all of the TWIVs are recorded on the road, which is fine. It's something different. I do miss the incubator. And tomorrow I will do another Beyond the Noise with Paul Offit. He wants to talk about respiratory syncytial virus vaccines, the 2 that have just been approved. So that'll be cool. And then we have a TWIM. And then at 5 p.m., TWIV clinical update with Dr. Daniel Griffin. So we have a great schedule. I love it. I'm having a good time. All right, so here, there was a good question at the top, but the person left and deleted it, so I guess I don't have to answer it. Yeah, see, it's a good question, but he or she took it away. Okay, so Vincent, I know your plate is full, more than full, but has the Amy papers been discontinued? No, they haven't been. I just haven't posted in a while. Amy continues to give papers, although the hose is less of a flow than it did before, interestingly. So I will resume. I want to do that because it helps me to sort my ideas for TWIVs and so forth. So, yep, I will be doing that. Let's see. Les had asked me a question. It's gone. Okay. Okay. Susan Weiss is the coronavirus expert. She's been working in coronaviruses longer than anyone else. That's correct. And the guests will be interesting as well. We're going to talk about SARS-CoV-2 origins, as I said, to try and discuss. I mean, we don't have a lab leaker on the panel, right? And it has been suggested that I have one of the lab leakers on, and that lab leaker has agreed to come on. And so I am thinking about it. I'm thinking about it. Because it's a very tricky thing, because there's no evidence. All this particular lab leaker says is that the Chinese have to do this, they have to do that, they have to give us this information, they have to give us that information. Okay, that's not happening, so where's your evidence? Well, they should give us this, and they should. That's not evidence. It's an absence of evidence. So here we have, on the other hand, evidence. So I'm not sure that is an issue. If we should cover it. I don't think we should cover it. Let's say hello to a few people. John, it's been a hoot going through the old Twivs that I missed. Yeah, the old Twivs are cool. It's 1014, 13. We have, 13. There are a lot of good stuff back there. Even the earliest ones were very naive, and that's a good word. We'll leave it at naive. They're fun. They're different. Different quality. Of course, they're just audio, but you can learn a lot. Noir, hello, Noir from Santa Fe, New Mexico. Noir loves Microbe TV. Oh, that reminds me. I have to order T-shirts for ASV. I don't have my, actually I do have my field notes, but I'm not going to reach down and get them. T-shirts for ASV. In the old days, I used to throw out T-shirts at a live Twivis. People got very excited. But I haven't done it in a while since the pandemic. I don't know why. I guess I got distracted. So I want to start doing it again. It's a really fun thing to do. Albert says, I listened to a TED Talk about how bacteria whisper to each other to mount coordinated attacks. Yes. We did this paper on TWIM a long time ago. Very interesting. Yeah, they produce chemicals that are diffuse out of them and go into other bacteria and say, hey, we have this common enemy here. I mean, they're not talking, but it's all about reactions. So what the viruses do, yes, there's some evidence, and we've talked about that on TWIV also. Actually, it was probably a TWIM because it involved the phage. A virus-infected bacterium secretes different chemicals than an uninfected bacterium, and those are thought to communicate with other cells as well. So the viruses themselves, of course, are not whispering, but they program the bacteria to make small molecules. The small molecules diffuse out of the bacteria, and they engage in other bacteria. The one with... let's see if I can remember what that was. I know it had beautiful, beautiful colors in it. So that was TWIM. What would you call it? I don't know. Bacteria talking. That's not going to work, I'm sure. Bacteria sing the blues. It was a really nice cover. No, I'm not going to find it. Of course, Bonnie Bassler has a TED Talk, How Bacteria Talk, and quorum sensing. She's very animated. If you want to see an animated microbiologist, check her out. Yeah. All right, what else do we have here? All right, so where's the question from Kip? Where is that question? Did you delete it? Because I saw it before, and it's associated with the little It's a little donate. Here we go. Here we go. Thank you, Kip, for your contribution to science communication. Kip and Laura Potter. Do you put any credence in the nicotine hypothesis, positing that SARS-CoV-2 competes with acetylcholine at nicotinic acetylcholine receptors, causing CNS neuronal dysregulation? No. So I don't think there's anything to it. Here's the reason. And I will share a screen. Where's the share screen thingy here? Here's the paper. An experimental test of the nicotinic hypothesis, which is what Kip just said, that these nicotinic acetylcholine receptors may be receptors for the virus, additional receptors. So what they did in this paper was to ask, can spike bind to these receptors? And so they produced the A7 acetylcholine receptor on intact cells, and alpha bungarotoxin is the competing ligand, and they tested different spike proteins and found none of them outcompete the bungarotoxic in a specific manner. We conclude that binding of the spike to the human acetylcholine receptors orthosteric sites, and thus its competition with acetylcholine, choline, or nicotine, is unlikely to be a relevant aspect of this complex disease. End of hypothesis, I suppose, right? It's pretty much the death knell. And as someone did say here, it would require that the virus be in the CNS, and we don't think it's there appreciably, right? So on top of the spike not binding to the receptor, it doesn't make a lot of sense either. Oh, and who was it that liked the title for Bird Flew Around the World? Oh, here's Les's question. Yeah. What do you think about India not teaching evolution in the periodic table? Well, yeah, of course, I think it's highly unfortunate as a scientist. If you don't believe, the periodic table is natural fact. It's the way the elements are organized, right? It represents the atomic beauty of the elements. There's no denying that, okay? Evolution, yeah, depending on your religion, what you believe in, but I don't think you should withhold information from people, right? Among all the Indian students, maybe there are some who want to become evolutionary biologists, right? Harmeet Malik is a fabulous evolutionary biologist, right? If they hadn't taught him evolution in school, he may never have been that. I think that's a shame to not do that. I understand that people have their views, but I don't think you should impose them on others, right? And that's a broad statement, of course, but especially with science, when we know evolution is clearly supported by data, there's no disputing it. And you may have your view of how the world developed and how many days, right? But you cannot have your own facts, and so I disagree with them not teaching evolution. That's unfortunate, but it also happens in the U.S., right? Also happens, unfortunately, in the U.S. All right, so that I had seen before, and I wanted to bring that up. And who was it that said? Oh, here we go. Peter said, "Great title, Bird Flew Around the World." So Alan came up with "I Flew Around the World." So I is the name of the patient from which they got the monoclonal antibody against the neuraminidase of influenza virus that's broadly inhibitory. And so that patient was I. So I flew around the world. And then the title that we had actually selected after the episode, and I'm going to show you how we do this. Watch. Let's go to the drive, and let's go to the TWIV archive, and let's go to TWIV 1013. I'm going to show you how this works. I think I've shown you this before, but it doesn't hurt to see it again. Screen share. So here's the Google Doc for TWIV 1013, right? And at the very top, we have the title ideas. And people are just free to put them in, right? And, these are all Alan Dove clevery things. And here is I Flew Around the World and Bird Flu, I Choose You, which I, again, is the patient from whom the monoclonal was recovered. And we all decided to pick that one. That's what the arrow means. And when I was finalizing the edit on Saturday, I said, I don't think people are going to understand this. And then I came across this incredible artwork of the chicken with a face mask, which I bought. I've never bought a piece of art, but I loved it so much. I bought it. And then I said, it's got to be Bird Flew Around the World. So I just took this one and put Bird Flew Around the World. And, these are the show notes, right, for everything. All the notes, the stuff that we have to go over, and the papers and so forth, and letters. But Kathy has lately taken to putting in purple here stuff she wants to tell us after the episode is over, so you don't hear this. But she said she was sitting in a diner, and the waiter came over and brought the check and said, the pot of coffee is yours. And she said, I didn't have any coffee. And it turns out he said, the bottom copy is yours. Check. That's an age-related phenomenon. Totally misinterpreting what somebody says, right? Okay. So that is the origin of the title, a long-winded origin of the title. By the way, now that I have remembered what else I wanted to tell you, here is a paper which we will probably at least discuss somewhat on Twiv. What's in a pandemic? Virus, disease, and societal disaster must be differentiated by Alexander Gorbalenya and Stanley Perlman. So Sasha, Alexander, Sasha, he was a student with Vadim Agol, who also trained Kostya Chumakov, whose position Amy Rosenfeld took when he retired last year from the FDA. And Kostya trained Eugene Koonin, all in Moscow. Amazing. So this is Sasha. I want to take another diversion. I'm doing a Twiv thing here. Instagram.com/profvr. I want to show you these people. It's so cool. Let's find my Instagram channel. Here we go. Okay, here. So I have to show. I am sharing already. So that is Kostya, and Amy and me. Look how tall the guy is. All right. So then we have Ann Palmenberg and me. And of course these are taken by who? Who took these photos? Yeah, Chris Kondayan, who did Twiv 1000. And of course is the University of Wisconsin, a well-known rhinovirologist. And then this is Alexander Gorbalenya. Sasha, really good bioinformatician, molecular virologist. Who else do we have here? Amy, of course, caught in an interesting moment. Here's Jim Hogle. Who's this? This is Lester Schulman. Or Schulman, I think. I misspelled it here. He is a virologist from Israel. And then there's Lester and me looking like, what are they talking about? And this is Mike Strauss from McGill. Anyway, I just wanted to show you Sasha. He's such a nice guy. Anyway, back to the paper that Sasha and Stan Perlman wrote. It doesn't define a pandemic. You might think from the title that they're going to define a pandemic, but no. What they're talking about here is we contend that using proper names for the virus, disease, and disaster acknowledges that they're separate entities representing biological species, organism, condition, and societal disruption. This recognition is crucial for untangling and communicating the numerous roles of humans and virus during pandemics, as well as for studying virus-host interactions generally. As we illustrate, using the example of the COVID-19 pandemic. All right, let me just pause here. Amy and I have been, and the whole Twiv team have been saying this for 3 years now, right? The virus is SARS-CoV-2, the disease is COVID-19. Don't confuse it. Now, obviously, we're not the only people who think it's important to name them correctly, right? But this paper codifies it, right? They make a formal paper out of it. Here's another paragraph that I like. The disease-centric concept is not compatible with recent advances in biomedical science. Disease develops independently in each infected individual who may also remain healthy. Furthermore, many viruses have been identified that don't cause disease. Thus, the application of disease-centric language to describing viral infections diminishes the complex relationship between viruses and diseases and is misleading. For this reason, having different names for the disease and the virus would make it straightforward to differentiate them. That's a very good point. I like that. So we talk about AIDS and HIV-1 and COVID and SARS-CoV-2. After 3 years of the pandemic, the use of COVID-19 greatly outnumbers SARS-CoV-2, especially at the science-societal interface. While this disparity could reflect reality, we believe it's largely due to the massive amounts of incorrect references to SARS-CoV-2 as COVID-19. Absolutely. I correct people all the time, and many people don't like being corrected. So let's see. Are you seeing this whole table? Yeah. So this is common, inaccurate, or ambiguous terms. So we have virus, right? SARS-CoV-2. Inaccurate term, COVID-19 coronavirus, COVID-19 virus, right? COVID-19 variants, reproduction rate of COVID-19. Then they have the disease, of course, which is supposed to be COVID-19, not COVID, not coronavirus disease. It's supposed to be COVID-19. Diagnostics, SARS-CoV-2 test, not COVID tests. Hey, I'm guilty of this. So is Daniel Griffin. Go get a COVID test. Are you COVID positive? That's wrong. But people don't want to say SARS-CoV-2. All right. I think people don't really. I think you get this, okay? They go on to epidemiology. So it's SARS-CoV-2 origin, not COVID origin. The vaccine, it's not a COVID vaccine because it's well, why? Why not? It's a COVID because it's got SARS-CoV-2 components in it, right? And they're not COVID drugs. Anyway, it's a good paper that codifies all of these issues that we've been talking about for a long time. And that will go in the Amy papers. There you go. I did want to point that out to you. Hello, Questella. How is it 8 o'clock already? Time flies. It does. It does. I am enough. Thank you very much. I appreciate it. It's 185 people feel that I'm enough. Thank you. I often think, I know what I know, but will you be properly taught by me? And obviously the answer is yes. So thank you very much. The blue glasses plus the dark background is a look. It is. I've been working on this look for 3 years. Let's start building this basement studio when the pandemic started, right? And I used to have the background brighter, but I kind of like this look. The only thing I don't like is, you notice the reflection of the lights in my glasses. There's a video controller here in front of me, which I can use to switch, my inputs. I don't have any other inputs here, but there is one. Well, here's, which one is it? Super something. I can't figure it. Anyway, it's reflecting in my glasses, and so is the white text. Actually, I can hide the white text, and I bet it goes away. Yeah, there it goes. That's gone. So now just the white look. The white glasses are there. Anyway, yeah, I like the look. It's different from the incubator, which is bright, right? Okay, it's not just you, it's just you. Remember at the beginning, I used to say, you're not just a student. You're not just a doctor. You're not just a nurse. You're not just a politician. You are what you are. I really believed in it very early on. I still believe it. You're whatever you are. So I am just me. Here's what you get. I love viruses. I'm crazy about them, and I want to teach you about them. That's what it's all about here. Hello, Lise from Columbus, Ohio, where next year, it's about a year from now, will be doing ASV. That should be cool. And here we go. This is the Venmo address at the upper left if you care to support us via Venmo. And welcome Viral Files and Micronauts. That's left from Mark Martin, who's a micronaut. Yes, Professor Mods and fellow virophiles. We had a discussion whether virophile was an appropriate thing to call ourselves. And apparently we have concluded that it's good. Hello from Coxsackie, New York. Slightly less apocalyptic than yesterday. I guess down in D.C., there was no haze in D.C., at least that at Samrid. I went in the morning, and it was fine. It was cloudy, but not yellow. There was no smoke smell. Yeah. Okay. Apparently it's been all over the U.S. It was a cool TWIV, yes. 2 hours, actually. We had a good audience, too. I didn't take a picture of the audience, unfortunately. Why? Because they took our cameras away to get in the place. They didn't want us taking pictures. But that's how I document my travels, right? I kept reaching for my camera to take pictures. And those were taken by, well, one is the TWIV, which is a screen cap, and the other was taken afterwards. But there was an official photographer who took a lot of pictures, and I'm told I will get some of them. So we'll share them when we do. For sure. Okie dokie, Kang. Does the flu virus hemagglutinin bind to any part of the ABO blood groups? Would secretors of blood group proteins be protected from infection? No, as far as I know, there is no interaction with ABOs. Let's see. Influenza, HA, ABO. Let's see. I've been reading about influenza viruses for ages, have never seen any association with ABO. And let's do one more search. Is there sialic acid on ABO? No, I don't. There aren't any. So there aren't any determinants of HA binding to ABO blood group. So people who have ABO, certain ABO proteins are not protected. No, I've never seen that. So Mike wants to know if N95s help with the pollution, which we've been having here. And Yvonne says we're being told to wear them outside because of the particulate matter. But we didn't have any problem in the D.C. area, for sure. Nobody was even talking about it. And yeah, I didn't even know it till I stepped off the train. Okay, in China, are they using Paxlovid and oral remdesivir for the many at-risk cases? I don't know. Now, Bully says probably Paxlovid, but I doubt Paxlovid, but remdesivir maybe. Let's look it up. Is China using Paxlovid? Paxlovid was added to China's national guidelines in March 2022. $340 per treatment. So it's approved in China. Yeah, it's approved, but it's not easy to get. As of January 2013, it was hard to get. I don't know what the situation is now. So probably not easy to get. Let's see. Is China using remdesivir? I guess they could knock it off, right? Chinese firm copies remdesivir. Chinese company BrightGene said it successfully copied Gilead's patented remdesivir, which is under clinical trial that was 2 years ago. So, they could copy anything. Or are they using it? Of course, they have their own homegrown antiviral. It's an old HIV drug, azuvidine. It's an oral antiviral. So there you go. It's not clear that either are being used in large quantities, right? Houston, Texas, which is where I will be in a little over a week. I will be there Friday, Saturday, Sunday, leaving Monday. A TWIV and a TWIM recorded on Sunday. So those will be released in the following weeks. Everybody's talking about the air quality. Yvonne says low confidence, not much according to the FBI. Yeah, I want the FBI guy to tell us that, right? By the way, Susan Weiss was on TWIV 609, a coronavirus chronology, and also TWIV 734, Weiss hath no furin like a virus scorned. That's a good title. Are there super masks with dual viral filtered inspiratory and expiratory valves? Don't know. I don't know. Does anyone know? So it'll be like a dual valve for inhaling and exhaling. Don't know. I want to leave it up to someone else to answer that question. And I'm sure we will find people here tonight who know the answer. K-ban. Can two different viruses co-infect the same cell at the same time? Yeah, for sure. It happens all the time. It happens all the time. I mean, that's how when you take the oral polio vaccine, right? You drink it, goes into your intestine, starts to reproduce in your intestine. And there are other viruses in your intestine that are highly related. Enteroviruses, type C entero or group C enteroviruses. And they infect the same cells, and out come recombinants, where pieces of the RNA among the two viral genomes are exchanged. Right? So the genome is a mixture of pieces from one virus and another. That's what we mean by recombinant. Happens all the time. Now, the recombinants occur with similar viruses, but a coronavirus is not going to recombine with SARS-CoV-2, at least not very efficiently. So if a coronavirus and a coronavirus could infect the same cell, sure, why not? But they're not going to recombine. I've never seen that happen. I don't think the mechanisms allow it to happen. The polymerases are not going to do that. But if you have different viruses, they can inhibit each other. We did years ago an experiment with poliovirus and adenovirus to see which would inhibit the other. And depending on the timing and so forth, you can get inhibition of one or the other or both. It's quite interesting, yeah. No, I'm not Woody Allen. I don't even look anywhere near Woody Allen, but maybe you didn't mean that, right? Is the Philly Twive open to the public? I don't think so. I think it's a university event. I don't have my email on this computer, so I can't look. So if it is, I'm going to ask Susan. I'll ask her tomorrow. If it is, I'll let you know. Mm-hmm. The spy agency's evidence is fun to read. Reports and blank reports, blank, blank, and blank. Moreover, sources say blank blank. On this is yeah, a lot of redaction, right? Yeah. How about a twick this week in Conspiracies with Debunk the Funk? This week in Conspiracies. Unfortunately, there's no time. We have two schedules we'd have to coordinate, and Dan's is busy too. Yeah, he's a real job. Not like me. I have 2 jobs, but there's nobody looking over me at either one. Nobody to report to, which I absolutely love. Right? I mean, I work for Columbia, right? I have a chairman who, and I don't know, he could tell me what to do, but he doesn't. Chairs usually don't do it. They can ask you to do things like be on a committee or something like that. But, when it comes to your research, the only thing they do is that they say you need to get more grant money. I don't have a lab, but he used to tell me that all the time, but I don't have a lab, so he can't fire me. I'm tenured, but, if I did something really egregious, I could be fired. But I'm not doing anything egregious. I mean, I don't think educating all of you folks is an egregious thing. I think it's a great thing. So I'm not reporting to him. And at Microbe TV, it's just me and a board of directors and all the podcast hosts. It's good. It's a good situation to be in. So anyway, Dan has to, coordinate with he usually likes to record on weekends because of that. Or late at night during the week after his child is put to bed. Anyway, there you go. In my former line of work, low confidence meant the issue had little or no impact. Okay. So why they release this and the press goes nuts and everybody thinks that it came from a lab as a consequence. So people don't understand, basically. I don't get it. Thank you. Margot says, thank you for these podcasts. It's so great to learn even a speck of what's going on. That's a great way to view it, actually. A speck. Yeah, it's better than nothing, right? But many people don't feel that way. They don't. They say, well, I have time to listen to these podcasts because they're just not interested in learning. And that's their choice. It's fine. It's just too bad. Twill this week in Lab Leak. Lab Leakers. I like to watch Dr. Wilson's funky videos in addition to Twix's. Yeah, it does a good job. One of the recent ones talked about Joe Rogan again, continues to spew misinformation. Of course, you have a very famous guy with Joe Rogan. So you put his name on your podcast, it's going to get you a lot of views. By the way, Alina, do you have a recommendation for a home microscope? I actually have my father's old microscope that he used to use in his office. And it's built like a tank. You just need an illuminator, but it works great. I'm happy to give it to you. I'll ship it to you. I'll pay for shipping because we were going to give it away. But if you would like it, just send me an email, vincent@microbe.tv with your address, and I will send it to you. It's got the mirror on the bottom to reflect the light up into the specimen path, right? And you just need to get an illuminator for that because we don't have one. It'll be cool. I'm happy to send it to you. My dad's, it was in his medical office so he opened his office in 1960. He probably used it before then when he was, studying. I actually have a collection of his slides of various tissues. They're so cool. Hematoxylin and eosin stained in the original wooden box with his scribbly handwriting, trying to identify them. Very cool. Tom says, it's tricky not wanting to give lab leakers credibility by association, whether how to engage, whether there are other urgent issues. So, No, I'm not joking. I will send you my microscope. I don't want it any longer. I'm trying to get rid of stuff. I'm trying to lighten my load here. It's just too much stuff around me. So Karen, my assistant, who's coming to the studio and coming on trips with me, she says I have to remove all this load of other stuff from my head and just have the science there. I never thought of that she said all this stuff is competing in my head and I have to get rid of it. So the same way I feel all the physical stuff around me. Like this desk is ridiculous. You can't see it, but there are no less than 1, 2, 3, 4, 5 water bottles. This is the latest water bottle I got from the Quebec trip. You can't see the writing, unfortunately, but it's a cool bottle. It's hard plastic. Université Laval. Here, I put my hand in front of it, and then we'll spin it around. Université Laval. See, and then we have the aquatic virus workshop. Right, it's on the other side, printed on the other side. It's cool, right? Nice water bottle. I mean, how many water bottles do I need in front of me? Then, one more I'll show you. This one I got at Tulane when I went. And they were very jealous. The National Regional Primate Research Center. This has got a cool lid, too. It's another screw-on lid. Yeah, cool. It's well made. And it's big, as you can see. I mean, it weighs a pound. So anyway, I have 1, 2, 3, 4, 5 water bottles. I have 3 glasses. I have a candle. I have a mug. I have tons of hard drives, hand cream connectors, like this kind of crap. Pens. Here's a Tulane pen. I don't want to get rid of this. Anyway, Lena, I'm not kidding. Vincent at microbe.tv. I'll send it to you. So the lab leakers. So this person is a Harvard professor who has been a lab leaker, but all she does is say we need this information. China needs to give us this, that, and the other. What's the point of that? That's not evidence. It's not evidence. I'm sorry. So I'm debating. I mean, I'm debating. I don't want to make it a debate. I don't think that this lends itself. We could just have a discussion. I can't understand why the health authorities won't give us all access to COVID antivirals and call it a day. Well, you can get Paxlovid now. It's licensed, so any doc can give you a script, and you could take it to Rome if you're going to Rome, right? You could just say, I'm going to Rome, I need a script for Paxlovid. They'll write you a script. They couldn't do that under EUA, but now licensure, it's there. You got it. Remdesivir, you can't get access because it's intravenous, right? You can't do that. People will be messing up their veins if you give it to them. But Paxlovid, Purevir, that's still on EUA, so you can't do that. Now, you want them to just pay for it and give it to everyone? I personally wouldn't have a problem with that, but, I'm not an economic genius, as Amy would say. I'm not an economic genius. I'm not a science genius, but I know my science. And, that's not going to work. Okay, I requested Paxlovid. Doc said my other medication is contraindicated. So he gave me Molnupiravir. I don't mean to sound silly, but how good is it? Because mostly I hear Paxlovid. Yeah, Molnupiravir isn't very good. It's about 30% effective at reducing progression to hospitalization. So it's not great. But you're still here, so you did all right, right? But it is the alternative to, because what's contraindicated is there are 2 drugs in Paxlovid. Normatrelvir, which is the protease inhibitor that hits the virus, and then ritonavir is a drug that makes the protease inhibitor last longer, and that can interfere with other drugs. So that's the problem there. We certainly could use more antivirals. We could use one without this ritonavir component for sure. People are working on it, but it's becomes an economic issue how much money you can put into something when the other antivirals have the market already. But again, that's not my strength. I could talk about the science. We could certainly make other antivirals. They're in development for sure. Yep. Nicola, I know your schedule is pretty full, but in the future, I think it would be nice to have Lydia Burubia from MIT. She is a fluid dynamicist specializing in biophysics of infectious diseases. Okay, Lydia, Lydia Burubia, Bourouiba, MIT. Okay, I write it down. Too many pieces of paper all over. I know my field notes. I should write it on that, but it's behind me. I don't want to get up and risk having the earplugs come out and make a mess and everything. But thank you for the recommendation. I think that's great. Do I have a spare centrifuge? I did 6 months ago when I, got rid of the lab. I could have given you spare centrifuges, but they're all gone. Sorry. Doreen, hello. Love the last Beyond the Noise or COVID mandates. Looking forward to the next one on RSV Vex. As usual, answering my questions before I ask them. Thank you. Yeah, but Paul is great. I love the short 10, 12 minute things, right? Explain things clearly. Should be viewed more. Why don't more people view them? Spread the word. Good evening. Are you still working on engineering polio as an oncolytic virus? Well, I'm not. I'm not working on any virus any longer, but other people are, yes. It's back to the drawing board because for glioma, it was not terribly effective, so they're rethinking it, and I'm not sure how much better they're going to get it, but they're working on it. Ever thought of a tour of a real lab showing the tools you use? Yeah, I did. I did a couple of videos in my old lab about plaque assays. Where are they? Did I ever publish those? Let me just look. Thank you for reminding me. The pot of coffee is yours. youtube.com/profvrr. And we prevent it from... Oh, good, it didn't. Oh, look, it tells me that there's a live stream going on. Cool. All right, now let's search within my videos. Where the hell is the search thing for my... There it is. Boy, what a crappy interface this YouTube is. Oh, my gosh. I can't search for my own videos. I want to search for the plaque assay video. I want to see if I ever released it. Search across your channel. Plaque. Oh, counting viruses, I think. Counting viruses. Virus Watch. Yeah, we did we did a plaque assay. We did a plaque assay. We did a... Oh, yes, here it is. Let me share it. This is the video. Virus Watch counting viruses. And this is just plaque assay. So check that out. Let me get the link here. We'll copy this and I should be able to paste it in. Yeah, look at that. On another computer entirely. I love how that works, right? You can paste it in on another computer. But as far as other things go, I would love to do that. I don't have a lab anymore. Maybe I could go to Amy's lab. Would you think she would do it? It's a good idea. Amy lab videos. We could do like pouring gels and doing PCRs and sequencing and all kinds of stuff. Any opinions at Samrid as the source of SARS-CoV-2? The people on the panel were all, it's not a lab leak, but nothing official. We didn't talk about it. Do you yet understand the mechanisms of the phenomenon of emergence? You mean like in general? We have some, there are some principles. And I think you need to go listen to my lecture on emergence. It's because it's, viruses are reproducing in animals all over. There are lots of viruses. They mutate every time they reproduce. So randomly, viruses emerge that have the ability to infect another species. And so then if you have contact between, say, a bat and a human, then the bat virus can get into the human, or the mouse virus can get into the human. That's the general idea. The details are in that lecture. And, we've watched a dozen or so viruses spill over from animals into humans, so we're getting it, although the details still need to be worked out. But those are the general mechanisms. You have a lot of population diversity in viruses in different animals, and then as the human population grows, impinges on animal habitats, or it forces animals out of their normal habitats, so they now go encounter humans, or we capture them and eat them, and in the process get in fact, it's all about encounter, and then, of course, once the virus is in a human, say, it has to reproduce sufficiently to get to another human. And you need to have a certain level of reproduction to be able to be shed and transmitted to another human, so it already has to be pretty good. And you need a chain of infection for the virus to spread. You can't have a lone farmer in a field somewhere get infected with a bad virus. It's never going to spread to anyone else unless that farmer contacts other people. So those are the principles. But the lecture is really a good summary of it. I would say, let me just find it for you. Emergence. So the two lectures, they go hand in hand. Here it is. I will get you the. We don't want to hear that. I'm going to get you the link. And we're going to paste it in. There you go. Okay. I would not like to be under a doctor who did not believe in evolution. I think I would not like to either. Have you tried using AI to name episodes? I have not. But almost everyone used it to solve the last TWIP. It was crazy. I know it gets the answer, but isn't it more fun to figure it out on your own, to be a sleuther, to be a detective, right? I think that would be a lot better. So what would you do? Give the ChatGPT some information, right? Why don't we try it? Let's try it real time here. I gotta have a bookmark. Here we go. Introducing Try ChatGPT. So I have to share my login. Okay, I have an account. Yeah, I have an account. Here we go. Done. I'm going to share this with you, and we're going to try naming an episode. I have to move this up a little here. We got to make it smaller. How's that? Okay, so we're going to start off. Could you make names for podcast episodes? Certainly. No, I don't want you to do it now. Look at this. It's giving me all these. I didn't. Now. Okay. Can you make a name, a catchy name for a podcast episode on H5N1 avian influenza spreading around the world. How's that? Feathered Fury. Unmasking the global flight of H5N1. What do you think about that? Let's regenerate it. Flocking Fear. Unmasking the global... Oh, come on. You used it. All right. So those are two. That's kind of cool. We could work with that, right? Really interesting. Thank you for suggesting that, NYC Cohen. That's really good. It's really funny. Peter, any idea how bacteria acquire viral gene sequences that they use CRISPR to destroy the attacking virus? Yeah. So when the bacterium is infected with a virus, the DNA is chopped up and pieces of the genome are integrated into the bacterial genome in the CRISPR array. And then those are transcribed to produce the RNAs that are the guide RNAs. Now, inherent in this explanation is that the bacterium doesn't die from the infection, right? So it has to survive. So not all bacteria survive a virus infection, but some do, and those are the ones that will have integrated pieces of the phage DNA. And the DNA is chopped up by endonucleases that are in the bacterial cell and then recombined into the genome. So that's where it comes from. Okie dokie. Antibacterial versus regular hand soap. Is antibacterial a necessity for hand washing? No, actually, it is not. What is in an antibacterial soap that makes it? What is antibacterial soap? What's in it? Oh my gosh, so many. What is considered antibacterial? Contains certain chemicals not in plain soaps. Yeah, I know that. What are the chemicals? FDA. Oh, here, the FDA says, I'm going to share this with you. You know, tonight is a real learning. So this is the page from the FDA. Antibacterial soap, you can skip it. Use plain soap and water. This is from 2019. Antibacterial soaps contain certain chemicals not found in plain soaps. Many soaps labeled antibacterial contain triclosan, an ingredient of concern to many environmental. Yeah, triclosan is an antimicrobial, and it's generating a lot of resistance, so you shouldn't use it. EPA regulates triclosan as a pesticide. Wow. Anyway, consumers keep washing with plain soap and water. That's still one of the most important steps you can take. So you don't need the antibacterial. Thanks to the FDA for that. Yeah, I hate to think what antibacterial soap and other antimicrobial products have on sewage systems that rely on live bacteria to process the waste. That's another thing. But we did a paper on TWIM not too long ago on this antimicrobial. So let's see, triclosan TWIM. What's that one? They just ignored the This Week in Microbiology. Just Week in Microbiology. Yeah, Petra Levin had done a project in her lab on induction of antibiotic resistance by triclosan. So the idea is that triclosan makes the bacteria resistant to other antimicrobials, not just triclosan. So it's not good to use it. But yeah, also the sewerage systems, that's quite interesting as well. By the way, Les does the timestamps for the TWIV videos and probably other videos as well, I guess. You do that? Let's see. Well, I think Twive 1013 bird flu. Yeah, he does Twive. He may do others. I don't know. But either way, thank you, Les, for doing those. We appreciate it. Are those speakers? Yes, these are speakers on my my left shoulder. Those are called magnaplaners. They're very big. They're thin, right? They're a film. The woofer is the big part, and the tweeter is the thin part on each one, separated by wood. Bought them years ago at a store in Manhattan called Lyric Hi-Fi. Since I think they're out of business, but I don't know. But they sound pretty good. But they're not being used. They're just blocking off the back, which is full of crap, and you can't see. It would look horrible. It's just junk piled up that I have to get rid of. Like I said, I have to clear my head. I have to clear my physical space. And behind those, I would like to sell them. If anyone would like to buy magnaplaners, I'll give you a good deal. If you want to buy a bridge, I can give you a good deal on that too. But I would like to get rid of these magnaplaners. And they work. I'll give you a good deal. They are speakers. Yeah, one problem with SARS-CoV-2, it's clumsy, right? So AIDS, HIV-1 is not clumsy, right? How about just CoV-2? Probably not good enough. Yes, I think that's part of the issue why people don't want to say SARS-CoV-2. I totally get it. COVID just rolls off the tongue. Yeah. There are lenses meant for exactly that purpose. Yeah, non-reflective, but they're not blue. They're just plain. The option would be to get rid of the video switcher, move it somewhere else. I could easily do that, or just cover it. Hey, that's a novel idea. Let's just try covering. There you go. Problem solved. That's the low-tech solution, right? Just put a piece of paper on top of it. Oh, my. And I don't need to push the buttons here on this live stream. No. In fact, I don't need any video switcher. I tried to get fancy with video switching, but never did it. Back to basics. Candle would be nice. Yeah, I have candles. But they're not lit. Yeah, here's one. Look at this one. This is very funny. Positive energy. A candle. I am a conduit for universal energy, allowing me to do good things. This is just a crock. Positive energy. It's just a freaking candle with a scent. But positive energy. People buy that, though. Sorry, I don't mean to be judgmental, because I know some of you probably like that, but it's crazy. Positive energy. I like the smell, but it's not, it's just the smell. Yeah, you would like a candle up there? That's another thing I have to remember to do. Okay, Polio Pete. Hello, Polio. I haven't seen you in a while. Question, can you ask Daniel if any hospitals in Queens and Nassau County are administering the 3-day remdesivir infusion on an outpatient basis? Okay, Queens and Nassau. When you come into the incubator with some whatever, Pignoli, Queens and Nassau County, 3-day remdesivir infusion, outpatient. I will ask him tomorrow, because we are recording the clinical update, as you probably heard. Good to see you again. Glad you came by. Tamid is from Bangladesh. It's 6 o'clock here. 6 o'clock, probably a.m., I guess. Yeah. 9, 10, 11, 12, 1, 2, 3, 4, 5, 6, 9 hours difference. Your lectures, advices are always encouraging me in my career. Well, that's great. I'm from all the way around the world. I'm happy to be able to do it. This is what I really like, being able to help people in all places. So you're welcome. It's my pleasure. The needle. The tour of the BSL-4. If you haven't seen it, oh man, that is really cool. Here, threading the needle. Nobody has gone in to a BSL-4 and done a tour, okay? And we did. So there were 10 years ago. Episode 68, well, it was TWIV 200. I don't know why there are two videos here. Well, this is the trailer here on the bottom. Right here. What is this one? Threading the needle. TWIV 200, we went inside, yeah. Anyway, 139,000 views isn't bad, right? That's pretty cool. So check it out. That's pretty neat. Why did I just bring this up? Yeah, because John mentioned it. The Hayes moved into D.C. today. So I left on a noon train, and when I went under the subway or the metro at 9:45, there was no Hayes. So it must have been after that. Yeah, I suppose it was a matter of time. Those of us from Ohio who are here on Wednesday night should meet up at TWIV next June. Yeah, you should. And we should all have a get-together at some point, yeah. Tuesday is the banquet, so I won't be available Tuesday, but some other night would be cool. This is very funny. I recently used one of my N95 masks to stain kitchen tables in the garage. This is very funny that you use a N95, which was so revered and so reviled, depending on who you were, to stain kitchen cabinets. Did it work well? I hope so. Is China still using ivermectin? Well, somewhere in the U.S., someone is using it. I can guarantee it. In some hospital. These are old stories. I'm not sure. Let's see. Let's put the date in. 2023. 2023. There's nothing recent out of China, so I don't know. Does anyone here have any information about that? There were at some point people who were in various places in China during this live stream, but they're not here anymore. When am I coming to Phoenix? Let's see at ASMQ. Let's just look up ASMQ. This is the Conference for Undergraduate Educators. That is November 17th through the 19th. Let me bring it up on the screen. There it is. November 17th through the 19th in Phoenix, ASMQ. Now, I know that you're not going to go, but we can have a meetup. If there are people in Phoenix, we can meet somewhere and chat. Bridget, hello from Indiana. Great to have you here. Martha, age 71, contemplating a 6th jab of Pfizer before a month overseas, hoping to avoid mild disease. Any data suggesting this might be an unhealthy choice? No, there are no data saying it's an unhealthy choice. If I were you, so I'm 70, I don't ride, what is that, a bull? No, you're braver than I. But I'm a year younger than you, and I would, instead of getting a 6th jab, I've had 3. I got COVID last November, and I immediately took Paxlovid. I was fine the next day. I would get a prescription for Paxlovid and bring it with you, because now you can do that. That now that it's FDA approved, instead of getting a 6th jab, because the Paxlovid will be with you, well, forever. As long as you have it, it'll work. Whereas the booster will only boost you for a certain amount of time. So, but there's no negative to the booster, no. All right, we're not going to talk about that. Is SARS-CoV-2 the first coronavirus that a vaccine was invented for? Well, people started working on SARS-CoV vaccines, many years ago after that outbreak, but they never got near people. So this would be the first one that vaccines were licensed for. Yeah, first coronavirus. Yep. Tom says, recombination reminds me, one, are there groups of viruses more or less inclined to co-infect cells? Okay, those are good questions. So are there groups of viruses that are more inclined to co-infect? Not that I'm aware of, no. I think all viruses should be able to co-infect cells. As long as there's a receptor for both viruses, right? So that both combined, yeah. I think that there's no problem there. So the second question, are certain cell types more easily than others connected? What do you mean by connected? Well, they form a sheet, right? Different architecture from, say, muscle or nerve cells, but they are all contacting each other. There's something about the intestine that seems to predispose to recombination. So multiple infections and recombination. But what it is, we don't know. Because sometimes you can get recombination in the human gut that you don't see in cells in culture. So there's something going on there that's based on the architecture of the tissue. We don't know what it is. Really interesting questions, yeah. Tamid, hello again. I have a question. Can HIV infect other animals? No, HIV is not. The only possibility would be, non-human primates or old world monkeys, but they are not infected. You need to use different envelope spike proteins to do that. So no, HIV is human specific. Yeah, so it's now. It came from HIV-1 the main group came from chimpanzee simian immunodeficiency virus. So the real question is, can HIV-1 infect chimps? I don't think it can. Yeah, it is. Apart from humans, the chimps are one of the non-human primate species that is susceptible to infection with HIV-1. Yeah, so the answer is yes. Can non-human primates, one of the non-human primates. Pig-tailed macaques can also be infected with a modified HIV strain that differs in one gene. So a few monkeys can be infected, but that's it. Good question. I always get questions that I've never heard before. That's the benefit of talking to so many people, right? By the way, we have 203 of you here and 171 likes. Please hit the thumbs up button to get more likes and more people listening to viruses. Education. We want to provide free education. I'm telling you everything about viruses that's up there. Everything. You just ask the right question, it'll all come out. Like this one, I just had never thought about it. But if you ask polio, polio only infects people. Doesn't seem to infect anyone else. Given evidence of vitamin D's role in enhancing immunity against SARS-CoV-2, why isn't it emphasized in COVID-19 prevention guidelines? You know, it's interesting because certainly Tony Fauci said, yeah, I'm taking vitamin D. I don't need a randomized clinical study to tell me that I should be taking it. I took it for a while because I was not getting much sun, right? I was inside all the time. But then I started getting sun, and I figured I didn't need it, so I stopped taking it. But I agree that it doesn't hurt to take it. I mean, it's good for other things as well, so you might as well take it. Why isn't part of prevention? That's a good question. Is it not part of prevention? You know, what are you basing that on? That's the question. COVID, let's search. Does COVID prevention include vitamin D? Yeah, plenty of studies. So NIH, as of September 2022, there's insufficient evidence. Where did the insufficient go? There's insufficient evidence for the panel to recommend either for or against the use of vitamin D for prevention or treatment of COVID-19. This is a report. I should show you this because this is exactly what you asked for. COVID-19 treatment guidelines from NIH, September 26, 2022. And there have been some meta-analyses and clinical trials on the use of vitamin D for the prevention or treatment of COVID. Most of them had limitations. And so down here, there's insufficient evidence for the panel to recommend for or against the use of vitamin D in the prevention or treatment. So I guess that's part of the reason that NIH doesn't consider enough evidence in support of it. This Week in Conspiracy and This Week in, what was the L? I forgot. It would need 100 monitors. Well, that would mean there'd be a lot of people, too. This Week in Conspiracy. I don't know enough about conspiracy. That's not my lane. I can't leave my lane because I yell at people for leaving their lanes, and I can't do that. It wouldn't be right. No, I can't do it. But it's very it'd be appealing to have a lot of thousands of people who come to a live stream. But I'm good with 200, 400. We used to have 1,000. You guys ask great questions. That's all that matters. And hopefully you get good answers. Thank you for posting your virology course. Good. I'm glad you like them. I post them every year, and I try to update them a bit every year. So you're welcome. It's my pleasure. I want to do them as long as I can. I'm not joking. All capital letters, Vincent at microbe.tv. I will pay for shipping. I'll hopefully pack it up nicely. And you will inherit my dad's microscope. So my dad came to the U.S. from Italy in 1951. He stayed, became a surgeon and practiced from 1960 to 1984 when he died. And this microscope has been kicking around. It used to be in this office, but it's not here anymore. So, yeah, if you give me that email and your address, I'll send it to you. Yep. Is there any evidence that nasal irrigations with hypertonic saline works for viral infections? So Daniel always says there isn't enough evidence that this is of much benefit. I mean, people do it for sure. I had a guy on a plane next to me who's irrigating his nose with some smelly stuff every, hour. But what? So I think. I think you would have to do it frequently, right? Because it's going to remove. Let's say in the best situation, it's going to remove the layer of virus on the mucosal surface, then the cells are going to keep reproducing, and within hours there's going to be more virus there. So, I've never seen a study where they properly looked at that sort of thing. COVID, nasal, irrigation. So according to UCLA, it may help and it won't hurt. Small studies may be helpful for people already infected. Well, what does that mean, could be helpful? Let's see. Saline gargle. Well, interest in these interventions has led to clinical trials. It's too soon to say whether or not it lessens the severity of infection. So that's the bottom line, in my opinion, that we don't have a lot of evidence that it would do much. It's certainly not going to hurt. So if you want to try it, I think it's fine. It's not saline, right? People do this for all kinds of things. But so it's not going to hurt for this. Yeah. Yes, I read that. Yeah. No, don't cry. It's okay. My pleasure. And yes, you should take a picture of yourself using it. No problem. My pleasure. I'd be happy to give. If you guys, do you want a 2-lane? You want a 2-lane primate center water bottle? I don't need so many. This is very cool. Yeah, send a picture of you using it. That would be cool. Questella says, if I had a microscope, I'd use it to look at these lice the kid keeps getting from school. Yeah. I remember the lice coming home for sure. Yeah. Pain in the neck. Pain in the head. Conference swag. Yeah, they gave me a bunch of swag at US Samrid. This leak in pipology. That's a cool phrase. I don't quite get what it means, but it's fine. Did you ever hear back from Andy Slavitt about him not acknowledging you guys? Oh, well, no, I asked his PR person, and she said, oh, no, he doesn't acknowledge his appearances on other shows. Oh, no. It's just crap bullshit. People have this stupid rule that they can't help others, which is all is anathema to me. It's the complete opposite of what I want to do. And I always encounter this every day, people not wanting to help someone else. They want to protect his golden egg, right? So, no, not happening. And I'm not going to pursue it. You should sell your stuff to the Twiv audience to raise money. So I do sell stuff on Cafe Press, but here's the problem. It's expensive to begin with, right? Like a mug like this, which is pretty cool. This is at $15. So how much can I add to make? I don't want to add $5. So I add $1. And, we make, in the first 2 years of the pandemic, I think we made $20,000 from, that's a lot of stuff people buying for $1 a mug. That's a lot of mugs and t-shirts. But I don't want to add more. However, I did get the information from the company who did the Spike shirts. And they're really good quality. And maybe if they could do them in mass, they would be cheaper and we'd make more of a profit. So I'm looking into that. There's all this stuff that, if I had an assistant, that would be what they would be doing. But no volunteers, please, because volunteers don't work. I have to pay you. Spike, no, not Spike, Twiv, T-shirts for ASV. That's the Spike kind. So anyway, hat, you can make money, but, the kind of money we need to make this a proper science communication company is not going to be had from T-shirts. Yeah. Is there any water in any of the 4 bottles? No. This one I actually, they gave it to us for lunch and it was full, and I took it home, so that was drunk. No. And by the way, I didn't show you this. This is a Columbia University mug full of erasers. These are my favorite erasers, by the way. Do you like these erasers? They're so cool. Polymer. I mean, I don't write with pencils much anymore. Polymer eraser. I'm putting it here because it's in focus. So there's that. And then this is the only water I have. This is full of water, right? The other bottles, I should fill them. But I don't know. There's just a lot of things to do before I start this stream, and I don't get to do it. A lab leak. It's impossible to prove something never happened. That's correct. But you can also have a lot of evidence that something else happened. And so then that negates the lab leak. And that's what we have so far, in my opinion. Are you going to ASM Microbe? Yes. Houston, Texas, next weekend, doing a TWIV and a TWIM on Sunday. Yes. And the TWIV will be with Eddie Holmes. It's going to be me and Eddie Holmes. I think that'll be a lot of fun because he's a funny guy and smart guy, too. I like him. Lab leakers should be in fact-free zones. Microbe is actual facts. I like the idea of a conversation about why they should still keep lab leak on the table and what kind of evidence we would need to see, but I hate when people say it was definitely a lab leak. I don't mind having a conversation either, especially when you highlight all the evidence for a natural origin, right? Yeah, but Robert Redford a few months ago when he testified said it definitely came from a lab. That's just irresponsible to say that. You can't say it definitely came from a lab. There's no evidence. Unbelievable. This guy ran the CDC for 4 years. As you all have no doubt heard, we have a new person nominated for CDC. Not sure I'm very excited about it, but we'll see. Craig says, can't they adjust your dosage of effective? Yeah, they can. So Daniel says you always have to examine what the patient is taking. You can sometimes stop the other drugs for a short period of time, or adjust the dosage. Yeah, but not always it works. Sometimes you cannot stop them, and so you have to find another approach. Yeah. What did Carl Sagan say? Extraordinary claims require extraordinary evidence. Yeah, but what are you referring to, Yvonne? The lab origin? I need to get Obsidian or a similar note-taking program. I like writing notes in little notebooks, right? So I usually carry a field notes, which our listener Jan gave to me a long time ago. But when I travel, I often don't take that backpack. I take other things, so it just doesn't always get taken, And if I put it in my pocket, then wherever I get undressed at night, that's where it's going to be, and I'm going to lose it. It's just tough. It's just tough to be Vincent. Ay, ay, ay. Maybe the new Apple headset will allow you to control a robot in a remote BSL training lab one day. Hopefully at the edu pricing. I don't know if it's good enough, right? If it's going to be precise enough not to make mistakes. So at the US Samrid, I got to look in the window of the BSL-4 lab, and see people working in there. I remember I did that in Australia. And the guy inside, he picked up a whiteboard. That's how they communicate. And he wrote on it. He says, "I love Twiv." That was so cool for him to do that. This guy working in a 4, thousands of miles away in Australia, Geelong. And he says, "I love Twiv." So, as I mentioned before, I'm hoping to do an Australia tour in September. And the plan is at least Sydney, but possibly Sydney, Melbourne, and Brisbane. Like a speaking tour. So stay tuned. Love to meet some of you. Okay, the gentle art of Swedish death cleaning. How to free yourself and your family from a lifetime of clutter. I wish I could copy this. See, I can't even copy the comments, right? Let's see if I do Apple C and then try and paste it into my notes. Let's see what would happen. Nothing. I can't copy this little text that's below here. Don't you think that should be a feature? I guess I could edit it. Yeah, maybe that's good. Copy. Let's try that. A new note. Okay, that worked. But now I have another place where notes from the live stream are being placed. So I have a little sticky pad here, right? Where I've made these are tonight's notes. And I have to tell you that this is a page from another live stream here. Here's something I wrote on the back of somebody's business card, and I have yellow stickies. Twitter poll: what is infection? Do you know how long ago I wrote this? December 2nd, 2020. Yeah, I never did it, but it's sitting here. And here's the other stuff. Here's an extra mouse sitting here. There are extra glasses. There are more notes. Daniel, vent in virus spread. Ventilation, T1000 T-shirts. Titrate. Oh, by the way, we're going to sell prints. Okay, that's on there. Guys, now I'm getting frustrated. Anyway, I'll read this book. I would like to read it, yeah. Yeah, death cleaning. That means when someone dies, you have to clean up all their crap. So when my mother died, I hired someone to just take everything away. I didn't hire them. I said, take everything away and you can keep it as payment, right? Yeah, it is brutal. I agree. Am I looking for a video editor for your shorts? It just sounds funny. I would like someone to make some shorts. Yeah, I have ideas about what we want to make shorts from. But yeah, if you have some experience, shoot me an email, vincent@microbe.tv. We'll pay you to do that. No volunteers. No volunteers. That doesn't work. Amy's lab is NIH. It's FDA. Yeah, I would need permission for sure. But we were there with a high school group last week, and we got permission to bring outside people in. But yeah, we would get permission to do that. I don't think shouldn't be a problem. I'm just going to... But you're right. Maybe they wouldn't like it. In which case, I'm sure I could find any number of labs of my colleagues where I could do that. David Quammen is updating his paperback to stress the origin of SARS-2 in the live market. Yeah, which it wasn't really a big stress in the original. That's right. Yep. He's working on a long magazine article for which he interviewed me. That's correct. I think the idea was the New York Times magazine. That's what he told me. And I wouldn't be surprised because he's published there before, and I'm sure they like his work for sure. Yeah. Yep. Could you do a Michael Faraday style Christmas lecture showing how virus research is done? I don't know who Michael Faraday is. All right. Another page. I just took off page 1. Michael Faraday. Folks, I'll do whatever you want. Was an English scientist who contributed to the studies of electromagnetism. Is that who you're talking about? Yeah. So it can't be. How could that be a video? Let me look at Michael Faraday video. I'll look into it. Sure. I wrote it down. Patricia is happy I'm back live. What do you mean? Because last week, I was with Amy last week, right? We did a Q&A. But I do have earbuds, but I don't need them because there's nobody talking to me. But I do like to hear myself and make sure it sounds good and all that, right? A lot of YouTubers are using Midjourney, etc. for thumbnails. I actually thought the masked chicken was AI art. I think it was. I bought it. I suspect it was AI art. What is Midjourney? Yeah, generative AI program. I would like to experiment with using AI to make thumbnails. Yeah, because right now I search for some art and that fits the theme of the episode, right? And lately I've tried to just have a visually striking image. So 2 weeks ago, we had just recorded the aquatic viruses TWIV in Quebec, and I was at the airport with Karen. And I don't know why, I just said, let me show you how I make a thumbnail. And then she found the image of the life preserver in the ocean, for aquatic viruses. And it was perfect. It must have been an AI image, right? The oceans with a little bit of horizon, and I could put aquatic viruses in there. And then the life preserver floating in the water. And then I found a virus and we tilted it and put it in the middle of the life preserver. So the two of us collaborating I really liked that. I thought that was very cool. But, we could generate images like that. I don't know. I have no idea how to do this. Love if someone wants to explain to me how it's done. So Sam, using Midjourney. Is Midjourney like a online computer? I mean, it's not a standalone app, right? It's something you need to do. You type in directions, right? You type in stormy ocean, right? With a virus in a life preserver. Look at this one. I was just looking at Midjourney, and this one is funny because it's the chicken. It looks like the chicken with the face mask, right? How would you use Midjourney? Like, what do you do? Do you just go online and do it? I assume so. But several people ask me if it was an AI photo, and I bought it from a company, and I assume it was, but I would like to make my own because it's not cheap, right? ChatGPT knows about twin, although it gets some of your bio details wrong. Well, it knows me. If you just say, who's Vincent Racaniello? It will give a good answer, actually. Isn't it from prior infection? Yeah, it is, but the bacteria have to survive, Vanity, right? Because if they die, it doesn't matter what CRISPR is in there, what array is in there. So that is the key, that some bacteria survive and they get the array. Found an article on bear bile, traditional Chinese medicine. Disgusting. Ooh. Well, you know, people do their thing. I'm electrostatic speakers, yeah. People do their thing, but they can't screw the world up from it, right? You cannot screw the world up, which is what happened with the Huanan seafood market. Huh, okay. I work under Dr. Farhan at RIT. She's a geneticist who works with VSV and C. elegans. I believe she would be a great guest. We both love your podcast. Maureen Farhan, RIT Twive. I wrote it down. Okie dokie. Now I will do my investigation. Speaking of bird flu, I take Tai Chi at a lake in Golden State Park. Steps away from geese, ducks, and seagulls, standing on feces occasionally. Should I be concerned? Yeah, those are birds that can have influenza viruses of all kinds, right? Well, if you're standing on them, I'm not too worried about it, but if don't get your face near it, right? And then the shoes you have to clean, and you might aerosolize it, right? I know when I was walking through the FDA, they have a lot of geese there, Canadian geese, and there's poop everywhere. You have to avoid it. I would avoid it. I don't want to scare you, but it is somewhat of a risk, so I would be careful if you can avoid it at all costs, yeah. Oh, here we are. Two-part question. This is the one I said in the beginning I wanted to answer. Gastrointestinal symptoms and fecal shedding of SARS-CoV-2 RNA suggest prolonged GI infection. Analyzed fecal RNA shedding in 113 people with mild to moderate COVID. They found RNA shedding in the first week in half correlated with GI continued 7 months. By what mechanism could the virus persist in intestinal cells for so long? Okay. So here's the story here. So when you have a respiratory virus infection, there's virus in your mucus. You swallow your mucus. It's part of your defense. It goes into your intestine. So with any respiratory virus infection, you're shedding virus particles in feces. These are probably not infectious. You can find influenza virus, rhinoviruses, SARS-CoV-2. You can find them by PCR, but if you did a plaque assay or infectivity assay, which nobody does, you would not find any infectivity because they're typically destroyed by the extreme environments in your intestines, like low pH and high pH and bile salts and enzymes of all sorts. So that's the source of RNA in poop, viral RNA in poop. But what about in the intestine? So this is fecal RNA shedding. I actually got this paper here. Let's take a look at it. All right. This is the paper, gastrointestinal symptoms and fecal shedding suggest prolonged gastrointestinal infection. I think the title is a bit too strong because all they have is SARS-CoV-2 RNA in poop by PCR. So they didn't look for infectious virus. As I said, nobody does because you need a BSL-3 lab. You need to be a bona fide virologist. And hell, I'm not a virologist, not me, but I'm saying the authors of this paper, we're not virologists, but we're going to do a virology study anyway and come to dubious conclusions. It's a good plan, as Amy would say. Here's the graph. Fecal genomic RNA measurements over time. So D, where does this darned thing continue? Fecal RNA concentration, okay? This is days, 0, 14, 28, 50. In the first few days, there's a lot of RNA. The first few days of the infection, yeah, you have a lot of RNA in your nasopharynx and you're swallowing a lot of particles, but then it goes really, really low here. 1 copy per microliter. That's what we have here. 1 copy per microliter. So it's really low, and I'll bet it's pieces, and it's not infectious. That's my opinion of that. Now, so nobody has shown that the virus is actually reproducing in the intestine. It's really hard to do. And I don't care if you do an electron microscope study and you find 1 particle in a few cells. Sorry, that doesn't tell me it's replicating. Apply some rigorous virology, please, to your experiments. What are you going to do in a person to show that it's replicating? It's really hard. And, you can make organoid cultures from the gut and show that it replicates. No, sorry, not good enough. An organoid is not a human. It's getting closer than a cell culture, but it's not a human. How do you show it reproduces in the gut? It's really hard. Poliovirus, you get a lot of virus in feces, and it's infectious. Nobody's looked for SARS-CoV-2. If you did and found a lot of infectious virus, then I'd start to listen that this might be a story. It's just RNA getting into cells, perhaps, and persisting a long time. We know that RNA viruses can persist in various tissues without replicating. That's a thing that apparently these authors aren't aware of. What could you do? So they're correlating the shedding with gastrointestinal symptoms. And the GI symptoms, it's an association, right? It doesn't mean that the RNA causes the symptoms. The symptoms, in my opinion, are due to inflammation, most likely. Cytokines can cause all sorts of GI problems and nothing to do with virus replication. Maybe there's RNA hanging around that's triggering inflammation, something like that. Now, if you thought it was replication, you could treat these patients with Paxlovid. If you had a cohort of patients who have GI distress after COVID, you could treat them with Paxlovid, but I don't think this is virus replication. I don't think that's going to tell you anything. I think it's inflammation. You can treat them with steroids, which you can do in the later parts of the inflammatory phase of infection. And then if the symptoms go away, that's what's causing them. You'd have to do a proper clinical trial to do that in a cohort of patients. But, I'm not convinced by any of these studies that just like there's no good evidence that SARS-CoV-2 is reproducing in the brain. I don't see any good evidence for it reproducing in the gut. And RNA can persist in various sites for long periods of time. We know that, as I've said. Okay. Let's see. We're getting near 8-minute mark here, so we need to start wrapping up. Are you going? Yes, I'm going to ASM Microbe. Am I a fully recovered audiophile? So, in the old days, I had big stereo systems with big amplifiers and preamps and big speakers. Still have a pretty big system upstairs with Van der Steiner speakers and Class A amplifiers, huge metal amplifiers sitting on the floor with a preamp. Never listened to it. Never listened to it. That would require me to sit in a chair for an hour and listen to music. Not happening. Listen to music a little bit in the incubator on my tiny powered speakers I use to listen when I'm editing podcasts, and I use those. They're not bad, but yeah, it's a bit different. There's no doubt that sitting in a chair and listening to music would be therapeutic, but I just don't do it. Fully recovered. I'm not an audiophile anymore. I can't claim to be one, though. Although I have the equipment, yeah, could do it. I will be by when the smoke clears. Unless you like smoked pinole cookies. No. It'll clear in a few days. Don't worry. Just let me know. Vincent at microbe.tv. I'm into the history of infectious diseases. Can anyone recommend a good podcast YouTube channel specifically on infectious diseases? History? I don't know. Someone else here probably knows. Lots of history media occasionally has infectious disease stuff, but I want more. There probably are some. Somebody will come up with it here. Let's see if we can find some. In our last minutes here. I was talking about, it's a perfect time to come to Phoenix, November. Yeah, it's not too hot. It's good temperature. Okay. All right. I'm happy to arrange the get-together in Columbus. Yeah. We'll arrange it. Get the so Kathy and Brienne are going to be doing the Twiv, so they'll be there. But Rich and Alan and others, they're not going to be there. But we can try. They're probably going to go to sessions at night, but I'm happy to play hooky. Yeah. Yeah, you can. We did an interview with Taubenberger, right? But we don't do much history now. That's true. What's the origin of poliovirus? Nobody really knows what animal it came from. Probably a rodent, possibly from Coxsackie virus. Don't know. Never going to know because you can't work on it anymore. Thank you, March 716, for your contribution to science education. Is there any specific academic text covering virology that you would recommend? Yeah, I don't know. What do you think I should recommend? What do you think I should recommend? I would recommend Principles of Virology. You can see them. Flint, Racaniello, Roll, Hatzianu, and Skalka. It's coming out in the 6th edition, but right now the 5th, two volumes, the best virology textbook on the planet, no doubt about it. What was the paralytic virus that Jane Goodall saw in a colony of chimps she was following? I thought it was polio. Yeah, she thought it was polio. Is Jane Goodall a virologist? Nope. No evidence that it was polio. Never got a sample, never did any infectivity assay, no PCR, no identification, nothing. Hearsay. And then people conclude it's polio because she said it was polio and she can say no wrong. But no, it wasn't polio. Wasn't polio. Are viruses alive? No, the virus is an organism with two phases. The infectious particle, which is not living, and the infected cell, which is a cell reprogrammed by the virus to make more virus particles. The particle is not alive. The infected cell is alive. So virus is an organism with two phases, non-living and living. Very straightforward. Is there a formal definition of what constitutes life? Yes, but many people don't agree with it. You know, something alive can you're defining life by what it can do, like metabolize, evolve, etc. And people say you can't use definitions for that. But I did once. I did make a video defining life, and Carl Zimmer got mad at me. So you can't define life by the properties of it. Hell, I can do whatever I want. I wrote the book. Rebecca said, is the bivalent vaccine for children going to protect kids from severe disease if they never had the ancestral strain? Well, the bivalent has the ancestral in it. Yes, it will protect them from severe disease. There's no evidence that it will not. One of these years, I'm getting that textbook. You never know. You might win it. Maybe we'll do a contest. All right. 9:59. Alina, no, you shouldn't give me $99 for the microscope. No, but thank you for donating it to Microbe TV. Now that I'm under pressure that the microscope is going to make you happy. Anyway, thank you for your generous contribution to science communication. You could open a Microbe TV store on eBay. Yeah, I don't have time. I just don't have time. I can't take on new things because I tend to take on things and then I don't have a lot of time. What did this here? I think Amy said, this is your gift to society. It is. This is my gift to society. And I want to keep on giving. Right? I want to keep on giving. Yes, 3D figurines. Virus figurines would be cool. Bobbleheads would be cool. All of that would be great. I need to find major TWIV donors. That's absolutely right. We have to find wealthy people who want to give us a lot of money because they believe in what we do. Now I'm going to skip all of these cool questions. I can't believe we have so many great questions, and I have to skip them over, and we're not going to be here for 2 weeks because, as I said, next week I'm in Philly. But 2 weeks from now, which is the 21st of June, 8 p.m. is Q&A with A and V. All right, so that is the next live stream. John wants to know what's in the lipid surrounding the RNA. All right, so the membrane around the RNA, the spike protein is in it, and then there are a few other membrane proteins as well. All right, everybody, I want to thank our moderators tonight. Who was here? Andrew was here. Steph was here. Tom was here. Vanity Nutrition was here. And Les was here. Give them all a big hand, folks, because they keep this a civil place. Thanks to all of you for showing up and asking all great questions. A lot of questions I couldn't get to just because there's not enough time, but I do appreciate you bringing them. You get next week off. Spring, look at it as summer break, but then we're back the week after. In the meantime, everybody, stay safe. Thanks for coming. Good night, everybody.